My name is Ulrike Reiss. I'm a pediatric hematologist at St. Jude Children's Research Hospital. I lead the Hemophilia Treatment Center here, and I have been the clinical PI on this clinical trial we've just published, and we're going to talk about.
I am Andrew Davidoff. I'm a pediatric surgical oncologist by training, and I'm the chair of surgery at St. Jude. But I also have a significant research effort and interest in gene therapy for monogenic disorders, and helped with the vector design part of the trial. So the trial enrolled patients with severe hemophilia. So severe hemophilia B, that means factor IX levels less than 1%, and factor IX is a critical protein in blood clotting. So these patients were very susceptible to bleeding. We produced in our GMP facility an AAV, adeno-associated virus vector-based gene transfer agent that was administered to the study participants as a one-time intravenous injection by simple peripheral vein administration. And then the patients were followed in this study for over 13 years, looking at the bleeding rates and the levels of circulating clotting factor IX.
The principal result is that the factor expression and the factor levels which were achieved within a few weeks of the initial vector infusion have been sustained throughout that time and have been durable up to 11 to 13, almost 14 years throughout the time we monitored the patients. So this is a significant finding, that we did not have any waning of the factor levels over time. Another finding is that, like Dr. Davidoff alluded to already, that the bleed rate decreased significantly and stayed down, which is somewhat related to the factor levels, but it's still very nice to see that even with relatively low factor IX levels that we achieved overall in the patients, that the bleed rate is very low at like 1.5 bleeds per year compared to an average of about 14 per year prior to that treatment. And 7 of these 10 patients came completely off prophylaxis. As you may know, or readers may know, that many of the patients with severe hemophilia B, or even some with moderate hemophilia B, have to use recurrent factor infusions once or twice per week or every other week through an IV infusion to protect themselves from bleeding. And 7 of the 10 participants could come off this completely and have been off throughout the 10 years or the 13 years of follow-up. Well, the other finding is that we didn't find any side effects related to the treatment, to the vector drug over these years. This is a relatively novel treatment, although this is 13 years out already, but it's unknown. There are many unknowns about what is happening in 10 or 15 or 20 years, and now we do have evidence of at least 10 participants, well monitored over 13 years, that their target organ, the liver, did not develop any problems such as a new liver disease or anything else. And that could potentially at least support, what is happening with other similar vector drugs, although it's not exactly the same drug, of course.
I think that the main findings for this study relate to the long-term follow-up of the participants, as I mentioned, over 13 years, and the significant finding was the activity of the replaced clotting factor lasted for the duration of the study without really declining over time, and that's the first time that long-term successful gene therapy has been achieved. It also showed the safety of the approach, and an additional point is that we found that the immune reaction to the vector itself was also long-lasting, suggesting that a second administration of the vector was probably not going to be successful. I think we've hit on the main point, that it is long-term follow-up showing persistent efficacy and benefit to the patients in the absence of any toxicity or side effects, and that the duration of follow-up is really what distinguishes this report.
This treatment has taken off a lot of burden of the disease for all these patients, all 10 participants. Even the patients who still continue prophylaxis have had significant benefit with reduction in their bleeding rate and reduction in having to treat with additional factor any bleed events during that time. So the effect on patients is significant as after having received that treatment.